As I understand, you welcome questions across the board.
Anything. Just, yeah, I mean, it's a small theater. I wish we could all turn our seats around and sit around in a circle or a square so we could have a discussion. So let's just try to keep it informal. Yeah, why don't we start right there?
Yeah, so in terms of trying to bridge that gap between the genomics and what you're saying, all these variations you don't understand, is there any thought, of course the difficulty is what cell types are used, but is there any thought of sequencing the RNAs so we can understand the functional differences?
Oh, absolutely. I don't mean to imply, I'd probably quickly say genome sequencing, but and one obvious place to sort of, if you want to see where lots of this is going on, is in the ENCODE project, but there's many others as well. So when I say genome sequencing, I mean the genome and the RNA in many ways, absolutely. And there's rich information, and by sequencing the RNA, doing all the fancy tricks to then sequence the DNA, but to be able to detect all the epigenomic marks on the DNA, as well as getting the variants, no question. And then you sort of alluded to it. Eventually, you're going to want to imagine doing this in individual cells and do single cell genomic analysis, both for RNA and DNA. All this is clearly of great value and lots of interest in doing single cell analysis. I'll tell you that much. Yeah.
Yeah, just two quick questions.
Yeah.
The first is about 2 years after the Human Genome Project came out, we had few others that followed, and being in the parasitology field that time, we had the malaria parasites.
Yeah.
Genome, and then we have the Anopheles mosquito genome.
Yeah.
And the whole idea is now we have the three. The soil is fertile to now power on to see what we can do. How much of genome of other organisms are brought into this project that you are heading? That's one. And
okay.
the second thing is...
By the way, do you want me to restrict my thing to infectious agents, or do you want me to talk more broadly?
Generally.
Okay, sure.
And then secondly, some of us are from the TGA, and you're talking about this, and I just sit here and think, okay, how is this going to impact our work as regulators of the outcome of some of these technologies? Because by the time you get to the area of personalized medicine, you're thinking of efficacy. How do you measure that comparatively to what we are used to, which is based on using large numbers of patients? So some of those things are just running through my mind that I thought, oh, is this really being considered?
Yeah, so I'm not sure I know exactly how to answer your second question, because I admit I'm a research scientist, so I actually don't even think about I think about the regulatory aspects. I'm not sophisticated to know how all these things might change the approaches you have to use in the regulatory world. That said, we recognize, it's part of the reason why that fifth domain is there, that we need to take responsibility as a research community to generate data to demonstrate the effectiveness, which is, I think, what the regulatory world is going to look for. They want to see document. And so, what?
Safety.
Yeah, and safety, of course. Right. So I think it's like any other medical advances. We need to be thinking about these things, being helpful when we can be helpful, and facilitate things the best we can. But again, I appreciate that there's a lot of complexities buried within that. In terms of your first question, what I would tell you is that there are many areas of growth in genomics, even beyond this vision of genomic medicine. Very broadly across the sequencing of many species. There's big efforts. For example, now there's even a named project called the Genome 10K, which is taking 10,000 species, carefully vouchered, the animal kingdom, for example, and then sequencing the genome, and because it's going to take years to collect all of them, and by the time those are all collected, the price of sequencing is going to be so ridiculously cheap, it'll be easy to do it. The idea is the future of evolutionary biology, I think, is going to be based on genome sequence exploration as much as anything else. You gave an example of infectious agents, and no question that genomics is changing the face of infectious disease research, and there's a whole institute at NIH that's dedicated to that, and they're spending huge amounts of money sequencing all sorts of pathogens, and we think that's great. It's probably not what our institute will focus on. It's one of these examples where that's probably good bets that we get the technologies and approaches out. Other institutes will pick this up for their areas of interest. Infectious agents would be, in that particular case, what that institute would focus on. So, you know, I think this is great that that's happening, and I wouldn't give the lecture, but I've heard the lecture given about how, you know, the genome sequencing will change the face of emerging pathogens, because the way they will monitor infectious agents in the future will be very much based on quick sequencing.
So at the moment, you're just focusing on the human.
Well, that's the, you know, I should also point out we're a small institute at NIH, we're only 1.7% of the budget. So we tend to think, how can we make the largest impact for the broader NIH? And we think about sort of these, notice I didn't really drill into any specific disease area. We think sort of enabling these major areas technologically, data analysis, and so forth is what our contribution could be.
Just,
yeah.
it's a bit of a segue then with, if you're only 1.7% of the budget, how do you actually focus? How do you focus the research? So you talked about translational research. So are there any criteria for, you know, focusing the effort? You know, are there any particular areas?
Yeah.
So that you because, you know, as you've highlighted, the data is just so enormous.
Yep.
You know, how do you
How do we as one institute sort of prioritize?
Yeah.
Yeah. So.
One to look at.
Yeah. So the short answer to your question is I have a very talented staff of extramural program directors, and we get in rooms and we debate all the time, just all the time, because we realize we can't do everything. And so we do and we talk to our advisors, and they all debate, and there's no right, there's no wrong, and then at the end of the day, you just got to decide. So what are some of the examples? I actually telegraphed some of them at the end in terms of what we see as the future. We, in particular, are looking for areas that we think will, if our institute, that would otherwise either that we're really good at, or that will otherwise slip through the cracks. So what do I mean by that? Well, we're set up now big centers to, you know, rare genetic diseases are classic things that tend to slip through the cracks, because some of them are multi-systems, and these, you know, debilitating disorders, maybe it doesn't neatly fit into one disease-oriented institute. And we felt that one of the things we could do is set up big centers, or really only medium centers, that could really industrialize this idea of taking ultra-rare diseases and finding the genetic basis. So that's a program. Newborn screening. Well, newborn screening also slips between the cracks, because when you screen for it, you can screen for all different diseases. No one institute necessarily stands up and says, I own this area. And except for in our case, we have a child health institute. They say we sort of own it, but we say we are involved in the technology. Let's do it together. So we partnered with them, 50-50. The clinical genomics information system. Well, wow, I mean, you know, sure, the Heart, Lung, and Blood Institute might be interested in those variants that are involved in heart, lung, and blood, but they're not interested in building a whole infrastructure. So we're trying to facilitate building it, and then we'll get other institutes to come in and help us curate that data around their disease area. So we constantly try to broker things. So, I mean, those are just several examples of things I talk. Pharmacogenomics, you know, that sort of also slipped through. No one, you know, everybody's interested in pharmacogenomics, but they're interested for those drugs relevant to their disease areas. So we feel this is an area we can contribute. So we're very, you know, we're strategic, but we try to, and in many cases we partner. The Cancer Genome Atlas, that's funded, so far it's funded 50-50 between us and the Cancer Institute. So we, you know, we put skin in the game and hope that people join us. Yeah.
Thanks. It's really clear that the speed at which the science of genomics is advancing, and you touched on the need for ethical frameworks around how we use this information. Do you think there's a danger that the science might out-accelerate the development of these ethical frameworks, and we may be in a situation where we look back and think, should we have done, you know, should we have done that the way we have, thinking of, you know, all this information that maybe things you wouldn't want, as a patient, you wouldn't want to know.
Yeah.
You know,
so
screening, do you screen for Alzheimer's, things like that?
So it's a great question, and it's probably a question we've been asking since 1988, before the Genome Project began. Is, you know, if what is this going to sort of start to come so fast? I mean, it's the reason why Jim Watson, when he was had my job previously, and he said, all right, we're going to dedicate 5% of our money to the study of ethical, legal, and social implications. The one thing we can do is to say we can try to get ahead of this by anticipating what the hard questions are, and simply getting research data to figure out whether or not, try to inform that discussion. So we are constantly looking at what's the next thing that might make us worried. I'll give you an example, and because we've been invested in this, we have a major consortium we just created around what's called a Return of Results Consortium. With all this genome sequencing going on, what are the obligations in terms of when you find, if you're looking for cancer, but you find something else, what are your obligations to return those results to the patients? And so that is sort of, some sort of unintended variance. And so we're studying this, and we're finding out what do patients want to know, what's the way to consent, how do you sort of work through that?
Do you go back and look every 5 years?
And then what's the obligation of a practicing healthcare professional? All these things. So we're studying this. I don't have the answers yet, but what I'm saying is we're seriously committed to it, to have a whole consortium. Probably must be about 8 or 12 investigators who have projects who are looking into this. Yeah.
Can you talk a little bit about research methods into the future? So particularly for the domains at the other end of the spectrum, so the science of medicine, the effectiveness of healthcare.
Yeah.
So I get the sort of the focus currently, which is around the other ends of the spectrum, but at that end, traditionally and classically, the sorts of tools we have are trials, clinical trials that look at safety, effectiveness, cost effectiveness, that kind of thing, and they're the planks that we use then for determining, what our health system would be interested in taking on. So I'm trying to work through then what are the implications of this in terms of research methods for that end of the spectrum, those domains into the future.
So I'm not sure I have a clean answer. In that fifth domain, we're really not quite doing that yet as our institute. So it's part of what we're trying to learn. I would say for advancing, there are studies that we are contemplating, even in that fourth domain, that start to ask questions. I'll give you an example of, if healthcare professionals had information about patients' variants that suggested they had increased risk for this disease or that disease, let's just start by studying what would they do with that information. What does that look like? Or in the case of pharmacogenomics, studies that say, okay, we believe this variant will indicate which medication gets person. Now let's do a large-scale study to say, there's studies like that going on now with warfarin, for example.
So it's the moving from the statistical associations you get with the really big...
Correct, those which are really discovery-oriented. Exactly.
And real populations
and
testing something.
Right, and there's two realities. I can say this as a physician. There's just two realities that are imperfect. One are healthcare professionals who are imperfect, and then there's patients who are imperfect. And you throw in some knowledge, and you wonder, how does that get translated? Do they want to use that information as healthcare professionals? The patients, do they want to hear this? Do they know what to do? So it sounds sort of like overly simplistic, but in reality, we don't really necessarily, we make a lot of assumptions as intellectuals what should happen, but we recognize that it's probably naive, and so we need to start to study some of the most simple things. Is that helpful? Is that?
I guess the answer is we're not clear, really.
Oh, there's no question we're not clear.
In terms of how this translates to those sorts of research methods for the future.
I think that's a fair statement, and I would also say, an area I'm not very sophisticated in, lots of discussion now around clinical trials, where should there be genetic stratification up front.
And so the numbers that you'd need to...
Exactly. Yeah. Although, yes, or you may get cleaner signals if you stratified up front by...
so what those stratifications are.
Currently, we're making reimbursement decisions in our healthcare system based on the results of those post-hoc subgroup analyses.
Oh, I see. Yeah.
Drug companies.
Yeah. That's right.
Find a drug, works on some people, do a post-hoc analysis. Oh, there's a variation here,
so they're the people,
and there's the claim. They haven't done a prospective study.
Correct.
Selecting patients on the basis against a control group.
Right. Or they feel that a trial has failed because they have a whole bunch of non-responders or negative responders, but if they only had done the genetic stratification up front, might that result have been different? That's right. Yeah.
In terms of the collaboration with the Australian entities, there is some specific domains that you want to implement with a higher intensity or proportions or everything is open, or you have a clear view of what is going to happen.
With Australian research? I mean, that's part of the reason I'm here visiting to learn more about this. Is just, I'm here to learn, and I think it's probably vice versa. Although it's not that it doesn't already exist, I knew before I got here, and I've learned more that Australia's in pancreatic cancer and ovarian cancers, participate in the International Cancer Genome Consortium, and we're participating in that. And so, we already knew there were places where there was common, interactions. And of course, I was in meetings most of this morning talking about possibilities of other areas as well. Yes?
On the ethical side, for a moment, you talked about newborn screening and that being charged with, many, many ethical issues.
Yeah.
Would you can you amplify that a little and how you're facing those ethical issues?
So sure, I could amplify a little that it's every time I mention in a room the notion of, should we be sequencing newborns, you hear people go, and I appreciate, and I talk to people and what seems immediately obvious to some, you recognize there's a lot of complexities. The short answer to your question, I'll give a long one, the short answer to your question is the way we're addressing this is saying we need a research agenda to inform this discussion, as opposed to saying can't, no, won't work. You need to sort of think about what the parameters might look like, and so that's what we're doing. And we have a call for applications right now, and over the next year we'll decide what we're going to fund. What we're envisioning is, how can you? To me, it seems. So I'll give you my bias. To me, it seems overwhelmingly clear that we seem to have reasonably good buy-in in most countries that are doing this for newborn screening for these, 20, 30, 40, depends what country you're in, for these 20, 30, 40 disorders. At least that, I think by and large, newborn screening programs have been deemed successful.
For the single
right. Okay, but that's based on genetic knowledge of rare diseases from 2009, maybe 2008. Okay. If we're going to see this number go from 3,500 to 5 or 6, at a minimum, we should see that number double, triple, quadruple, many more disorders. Okay, that would make sense to screen for.
But they may never actually. All of those disorders won't necessarily eventuate in their life.
No, well, there'll be but there'll be many that there will. Just like that's what we have buy-in on my first 20 or 30. Yeah. So let's just say, and at some point when you get to let's say there's 80 disorders, at some point, just at a practical level, it's going to make far more sense just to sequence. It's going to be more cost-effective just to sequence the genome than to do 40 independent tests. And once you've done that, for practical reasons, you now have this body of data. And you've got to figure out what you want to do with it.
Exactly, and what the newborn that's had no say in this.
It's right. Well, the newborn had. That's right.
Want to live their life thinking they may get this disease.
Sure. No.
Diseases that they can't do anything about. That they're going to get
and maybe at the end of the day, the answer is that we don't sequence the genome of the newborn, but we'll see. I mean, and I don't know. But so I think there's some fundamental questions about how to practically approach this.
There's increasing pressure in the private sector in Australia to start doing genetic or genomic screening of prenatal, even pre-implantation in IVF environment.
And by the way, do people, I don't know how many of you geek out like I do on genomic literature, but you realize that, you do realize that there's been, in the past 6 months, the past 2 months, there have been these incredible publications that describe how we could now pretty reasonably sequence an unborn child's genome simply by drawing blood from mom and getting out the DNA and fetal cells from mom and so I think we're going to see whole new approaches for prenatal screening. And could you even imagine then even actually truly routine, and even in some cases, I mean, it's way cool technically to think of it, but it's also, wow, that brings up a whole bunch of issues.
Newborn screening brings up issues.
Exactly. Oh, you only put it on steroids by backing it up. And I realize that, but it's not hypothetical stuff. So. Yes?
I guess it segues a bit into intellectual property and patenting and ownership of data.
Oh, speaking of easy issues. Yeah, okay. All right.
Yeah, of course. So I just wondered, I have been involved in a conversation about who owns the blood spots for newborn screening.
Yeah.
That has been a topic of conversation.
In the United States too, yes.
The ownership of those things that get taken when you...
The Guthrie cards, yep.
So I just, and the stuff you're talking about, just magnifies 1,000 times.
Yeah.
Your thoughts?
My thoughts, that's why I showed the slide where I said genomics and society. Under that, we're dealing with patents, we're dealing with Guthrie cards, we're dealing with, newborns. I mean, there's a whole host of return of results right on down the line. you know, it's sort of the, you know, there's a common thread here, which almost goes back to what one of the early questions was, is that, you know, all this is happening technologically. We're victims of our own success. These advances happen so quick that, you know, what maybe once upon a time the patent system made sense to some extent, you know, now it just doesn't seem to make sense at all with what's happening in genomics. And so, you know, and all these other issues, again, they're accelerating the complexity of the issues because of these technological advances.
I mean, I think, you know, there was a template perhaps with the BRCA genes, the patents and, you know, the story around the templates of the BRCA gene. And, you know, and how, you know, with the difficulties with getting access to testing for that. And so maybe that is a guide on how you might...
yeah, although I'm not sure everybody would agree. Yeah, although there's still controversy on whether that's a good example or not, but we'll see. We'll see. Yes?
I mean, touching on that ownership and accessibility to information, I mean, if you are planning on screening newborns or even pre-implantation type things, and then that person 30 years down the track wants access to that information, or their health insurance company, I mean, have you got ethics groups discussing?
Absolutely. Absolutely have.
And I mean, just merely the accessing the information is difficult for those who own the information but don't have the scientific knowledge to interpret it carefully. That's where we've come with individual people getting sequence information through those kits.
Which, without a healthcare professional involved. I mean, and that's one of the things that scares me about direct-to-consumer testing. Exactly that. I mean, on the other hand, you know, again, I think you could sort of paint, like many ethical dilemmas, you can paint different pictures and some people respond to it differently. You know, the one view is, wow, take that newborn blood spot, sequence that genome, you know, put it in their electronic health record, find out from those parents, you know, what they want to know or not know. Maybe they'll just want to know the 100 things which they could do something about. We do that in the next even 10 years. There'll still be huge amounts of lack of understanding what the genome sequence means, but maybe at the annual physical that child gets it every year, they check all sorts of blood work and they check the heart and they, you know, do whatever, but they also run that sequence through a suite of analyses that reveals whatever the latest information is. And I'm sure we'll still constantly be learning that part of your clinical care might be a reinterpretation of your genome sequences, that knowledge advances. And I could paint a picture, that sounds wonderful. Maybe some people want it, maybe it's not wonderful and some people will hate it. But, you know, what does that look like? I mean, I think there are some, you know, I'll give you another example. And again, it's a recent paper tragedy strikes, you know, the rare cases, I forgot what journal it was published in, where ultra rare variant in some children that got coding, just innocently were given coding at a very young age and it killed them. And I don't know, if I was a parent of that kid and you could say, I could look up early on in life what medications your child should never get, I would buy into that personally. I would want to know that. That would be on the list of things I'd want to know. So that child, and I wanted their electronic health records, no matter where they are, you can get to that electronic health record and say, don't give this child codeine or don't give this child this medication. And but we need to study that. Am I a rare exception, or is that going to be a more common desire of parents? We'll see.
I mean, to me, one of the challenges is in these early stages, we're coming up with some genes which are associated with diseases.
Yep.
But we don't know what other genes are affecting it either positively or negatively.
Right.
So I suppose it's a matter of what stage do people get someone who found a gene for a disease?
Right.
And therefore what are the modifiers, and until we know what modifiers are, we sort of seem to be in a really awkward situation.
So I do think we're talking about two different things here, and I think you raise a good point. I mean, on the one hand, some of the examples I'm giving are single gene, absolute causation, no question. That's what you see in newborn screening, that's what you see in pharmacogenomics. Then what you were just referring to are sort of these little statistical associations, and that's actually what these direct-to-consumer genetic testing groups are doing, which is why they're so controversial in some quarters, because they're just a sliver of the first information, of which the complexities there are so much to come that, you know, we don't want to overinterpret that. So, and you know, I think we see these boundaries already. I think they'll blur with time. But you're right, some of these things are much more definitive than others, and do we want to worry people unnecessarily? But, you know, on some things you probably do want to let them know what are absolutes. So, yes?
You've talked about sequencing DNA and RNA.
Yeah.
The obvious next step is to catalogue proteins.
Yeah.
Is proteomics in this grand plan at all?
The short answer is sure, it can be. Why not? As could be glycomics and metabolomics and all these other. I guess what I would say is, we sort of have to. I don't want to be accused of being unfocused, so our institute tends to have expertise in nucleic acid work. That's what our technology base is, and so that's where our strength is. I think the further you get away from nucleic acids, it gets away from our expertise. I think there's others that can pick that up. If you doubled my budget, I would take it on, but so I mean, there's a practical reality, and we've been asked to sort of help get involved in some proteomics things because we know a little bit about how to foster technology development and big data issues. But I would say that we don't have very strong ownership of that because I think it gets us a little too far away. But of course, all of it's being coded in the genome, which we care passionately about. Yeah.
Historically, the Human Genome Project started just when the rise of non-coding RNAs happened, and you need the sequence to search for microRNA targets, etc. I always expected LNA blockades and those kind of small RNA treatments to really take off, but I haven't really seen much. Do you have any feeling on how progress on that's happening? Like, for example, I know that there are experimental drugs to block, say, some of the microbes involved in fat metabolism.
Yeah.
But I've not seen anything clinically, really. Do you think that's a big expanding area? Or?
I don't know. I mean, I know lots of activity in that area in the biotech arena and to some extent in the pharma, but, I don't know. Are you not hearing about the newer trials because they're failing at the earlier stages, or is it just harder work, or are they coming? I don't know. I honestly don't know. I don't think it's a dead field by any means. I mean, I'm quite sure there's going to be some exciting things there. I just don't know how close they are to fruition. Yeah?
Yeah, I think in lots of disease, for lots of disease, not only the genomic, contribute to disease, but also the environment factors.
Yes.
Also contribute a lot, actually. Whether in your project would have been considered.
So the role of environment.
Yeah.
So here's what I would tell you. And in a longer version of this talk, I even have a slide to describe it. Clearly, environment is critically important. I alluded to it, especially for complex diseases, critically important. Why is it that most of what I talked about was genomics? Well, first starters, because I'm a genomicist, so that's what I'm going to talk about. But the second thing is because what has happened in the last 10 years has been huge surge in technology. Technology will drive all this, by the way, it always does. Huge surge in technology for nucleic acid analysis. And I have great friends in the institute that studies health, environment, and issues at NIH, but they will be the first to admit that they need a technology surge. It's probably coming. There's a lot of indications that there's new fancy methods for measuring environment and using cell phones to monitor and various electronic devices to gain exposure data and all that. But they don't have that surge, so they're lagging behind. I'll give you an example, though, where I really think, again, some low-hanging fruit for the future. And we stumbled into this in genomics because we had a technology to contribute. What am I talking about? I'm talking about microbiome research. So everybody's familiar with microbiome research. Our bodies are just filled with all these microbes. They outnumber human cells 10 to 1, 10-fold more microbes in us and on us than human cells. And these same sequencing technologies that can be used for sequencing human DNA can be used to inventory the microbes that are in us and on us, because you can just grab some specimen and just purify the DNA and sequence it and inventory what's there. By the way, 90% of the microbes that are in us and on us cannot be cultured in a laboratory, so we're seeing them from the first time by doing this DNA sequencing. And finally, the final point, microbes, our most intimate environmental exposure. I mean, and if you follow the literature, it's exploding right now. Microbiome research. It's clear that the microbiomes in the gut of obese individuals are different and microbiomes of individuals with bowel disease is different than normals, and just go on and on and on. I think you're going to see it. Why are you seeing a huge surge in microbiome, which is environmental exposure research? It's because the technology happened to be lucky. It could use our sequencing technology. But why? It's because the technology advances gave us that. So I think when a similar environmental monitoring for some drug or some chemicals or something or other is available, I think you'll see a surge in that too.
Eric, one of the very helpful things you did this morning was talk about how to keep in touch with what's going on at your institute. Could you speak a little bit?
Sure, happy to. I don't mean to be self-serving about it, but people were very interested to sort of knowing how could we keep updated on what is happening at, and I can only speak to my institute, but there may be similar ways at other institutes, at NHGRI, just so you can know what's the latest thing we're thinking about even, or what are the latest workshops we might have, or the latest new grant initiatives. And it dawned on, and and obviously you always would be welcome to email us, or you can, it's a long flight to come to DC, as I know. But increasingly, we believe in transparency of what we're doing. We really we have a community that's very interested in being engaged with us, and we're very open with them, and it's, again, part of this culture we have. And so, especially since I became director, and because these technologies are better and better, we put a lot of stuff up on the web as video. And in particular, we have, I know all of you are involved in various agencies that have advisory groups and so forth. I have an advisory council that I meet with every 4 months, 3 times a year. And it's a big 2-day meeting, and half the time is spent behind closed doors talking about grants and all the secret stuff. But half of it is what's called open session. And that open session begins with me making an hour-and-a-half presentation, 100 PowerPoint slides that my staff spends months preparing. But it goes through in gory detail everything we're thinking about, everything we're doing, and including in the field. I talk a lot about what's happening in the field. And I started video casting that live, which would be an inconvenient time for you. But we also video archive it, and it's just, we have it there forever. And it even includes my PowerPoint presentation. Similarly, all the presenters, and we typically have 5, 6, 7 presenters, other people presenting the latest things in this, the newest ideas. Even seeing the debate around newborn screening was an example, where we talked about that concept of would we go forward on it, and we did it in February, and they argued it down. And we came back in May, and they argued it up. So you could even watch that debate in instant replay. And we put all that up on the internet, and you could just get to it from our website and see it. And so it just suddenly occurred to me that if you were interested in hearing what was going on with us, and you just wanted to just spend a few hours every 4 months looking at stuff, we make it all available. And so you'd be welcome to do it. In fact, I would be delighted to know people were actually looking at that stuff and helping it be helpful to you in your planning in genomics research. So this has been a great conversation. I've really enjoyed this. So thank you.
Could you join with me in thanking Eric Green?